Hello, my name is Casey Moore, and this is my final presentation for Bio 103. I chose to research CRISPR Cas9. What is CRISPR? It stands for Clusters of Regularly Interspaced Short Palindromic Repeats. It is an inexpensive tool that allows easy manipulation of DNA sequences by changing the malfunctioning sequence to correct mutations that may otherwise cause diseases. Scientists are super excited because what it does is recognize the DNA sequence that is malfunctioning, then disable it by cutting the DNA or gene editing. Then they take an altered RNA sequence that matches the DNA sequence to be edited and put it into the cell where it finds the malfunctioning DNA and replaces it. There is a mechanism called protospacer adjacent motif, or PAM, which is a tag that the Cas9 protein must see in order to cut into the DNA sequence. It is a base pair DNA sequence immediately following the DNA sequence targeted by the Cas9 nuclease. It ensures that it will not edit where it should not. So simply put, the scientist damages the DNA by cutting it, and when the natural repair mechanism kicks in, they make it use the edited RNA to change the DNA sequence. What are some of CRISPR's intended uses? Currently, some of the things that they have been able to do using the gene editing technique is create mushrooms that don't brown as easily. They have used it to reduce the severity of genetic deafness in mice. And they have also edited bone marrow cells in mice to treat sickle cell anemia. And what they hope these advancements will lead to is treatment in single gene disorders such as cystic fibrosis, hemophilia, and sickle cell disease. And hopefully in the future, it's showing promise to be able to treat more complex diseases such as cancer, heart disease, mental illness, and even HIV. Now, there are many ethical questions about gene editing. When the cell that is being edited is a somatic cell, it only affects that specific tissue, and those changes don't get passed down to future generations. However, germline cells do. So that sparks fear and brings up questions regarding the permissibility of using CRISPR-Cas9 in germline traits, such as height and intelligence. A specific example of the ethical dilemmas could be scientist He Jinkyu, a SUSTech biophysicist who lost his university position and leadership of a biotech company that he founded because of the first gene-edited babies. He was called China's Frankenstein and stupendously immoral for producing an IFV baby naturally immunized against AIDS. He believed his CCR edit, making the embryos resistant to HIV, smallpox, and cholera, was going to be the biggest medical breakthrough since IFV was awarded the Nobel Prize. Some of the criticisms are that he is changing mankind or humankind. The fear is that he will try to use some of these technologies, or other scientists will try to use these technologies to enhance things like intelligence, change their baby's skin color, and increase sports performance. Now, Mr. Jinkyu has consistently rejected these goals, but with uncertainty brings opportunity for irrational fears or outlandish ideas. Some people have even talked about the idea of bringing back the woolly mammoth from extinction. Now, with any great scientific advancement, you will find resistance, and in this case, fear of changing humankind by creating designer babies. These fears should not overshadow the exciting work that shows promise in eradicating many debilitating and deadly diseases. Thank you very much.